Okay, these slides or this lecture overview is for metabolism, which is chapter 7. We'll start with a broad overview of what metabolism encompasses and then move into a discussion of enzymes. And this is one of your key concepts, is understanding how enzymes are regulated in a cell. We'll follow this by looking at the general forms of energy, specifically looking at ATP versus redox reactions. And basically we're looking at like the ingredients of how the cell metabolizes. So that this will lead us up to looking at pathways, which will be in the next sets of slides. Now, just to forewarn you, I'm going to try to do something a little bit different with these, with this particular video, and sort of toggle between these lecture slide overviews and then a kind of a projection of a whiteboard so I can outline a couple of these major concepts here. So we'll see how that goes. Again, there are two major sets of key concepts for this chapter. The first is covered in these first sets of slides, which is enzyme structure, function, and regulation. And the second will be looking at those metabolic pathways, specifically understanding the pathways involved in aerobic respiration and fermentation. So what is metabolism? Essentially everything. So all biochemical reactions that are happening inside that cell, physical workings. So that means the stuff, all the reactions that biosynthesize, so making larger macromolecules out of simpler ones, building those macromolecules into the components of the cell. And then also the opposite of things, bringing in nutrients and breaking them down into their individual components for the purpose of producing energy or producing those precursor metabolites that are used for biosynthesis. So we use these two terms, anabolic and catabolic reactions, to describe these two broad categories of metabolic reactions. So anabolism or anabolic reactions are those that are involved in biosynthesis, so the synthesis or the development of the creation of life. So these are all the reactions building up the cell components. And so because this is a building up or forming bonds, all anabolic reactions require the input of energy for those bonds to form. This is in contrast to catabolic reactions or catabolism. This is bringing in nutrients for the purpose of producing energy. So this is where bonds are broken, breaking down large molecules, again, for the purpose of producing energy, but also, really importantly, to produce these things called metabolic intermediates that are used to essentially provide those basic components for biosynthesis. This is a super simple picture of something, of a cell, and both of these catabolic and anabolic processes. So you can see how they're not separate processes. In fact, one is really required for the other. And this is, again, a super simple picture. We'll see a little bit more of a complex picture later on of this concept here between catabolism and anabolism. But in general, just kind of imagine the cell brings in energy in the form of an organic compound like glucose, which is a really common starting point for energy production. Glucose comes into the cell and then through a very specific process, and we call this the central metabolic processes, through glycolysis, Krebs cycle, electron transport chain. The end result of that breaking down of that 6-carbon compound is waste. So there are some acids through fermentation or carbon dioxide through aerobic respiration that are generated in this process. But also note that there is energy released as this molecule is being broken down, and also precursor metabolites here that can be used in these biosynthesis reactions. Here on this side, anabolic reactions, there must be some kind of input here, even with these precursor metabolites that are generated from breaking down nutrients. There are other nutrients that are needed, like nitrogen, phosphorus, sulfur, etc., that come into the cell, combine with these catabolic reactions, these precursors that are generated here, and then with the input of energy, these bonds can then be formed to make our individual subunits like amino acids, nucleotides. Those then come together to form those macromolecules. If you recall, we talked about this from the last chapter, the 4 basic macromolecules that make up every living thing. Proteins, actually, let's say the 4 basic, I don't know why it's kind of separated like this, but amino acids go into proteins, nucleotides go into nucleic acids, lipids go or fatty acids go into lipids, other types of lipids too. And then carbohydrates, so sugars, right? And sugars go into oligosaccharides or polysaccharides, or different types of carbohydrates. Anyway, so these macromolecules then form together the cell structures like cell wall, cell membrane, etc. And so each time there is a building up of larger, more complex structures, there's a requirement of the input of energy. This picture is from your book. I don't like it so much, but it does show sort of the relative complexity of each of these molecules that are either brought into the cell or developed within the cell to make individual bacterial growth. You can also imagine, instead of just saying the complexity, you can say the potential energy that these molecules hold. So here, for example, is glucose that comes into the cell. This is brought in from the outside. This has 6 carbons, 6 oxygens, 12 hydrogens, so it has a lot of potential energy. It's a complex molecule here, and through the process of catabolism, breaking down, and here again are the central metabolic pathways that I mentioned before: glycolysis, Krebs cycle, electron transport or respiratory chain, and alternately the process of fermentation. These things will completely or incompletely reduce this molecule, so break the oxidation, reduction, reduce. Oxidize this molecule, so breaks the bonds here, and releases energy as these bonds are broken. During this process, there are precursor metabolites that are generated, so pyruvate or pyruvic acid, acetyl coenzyme A, glyceraldehyde 3-phosphate. These are 3 examples of many, many precursor metabolites that can be generated through these catabolic processes. And these are either fully further oxidized to generate more energy, or they can be pulled out of these catabolic processes and put into these biosynthesis processes, which are anabolic reactions or anabolism. And you can see that the relative complexity, this is a pretty simple 3-carbon compound, is a 2-carbon compound here. These then are built up into larger components like amino acids, sugars, nucleotides, fatty acids here as your building blocks, which then get put together to form your macromolecules that then form the whole cell, right? And some are spontaneous reactions, but almost everything is actually, we're going to just say everything that's a biosynthesis reaction or a catabolism reaction in the cell is powered by a unique enzyme. This concept here, metabolic pathways, is just to give you a visual picture of how the cell works. So this is biochemistry basics. Within a biological system, specifically inside of a cell, chemical reactions, like you learned back in basic chemistry, don't occur independently. They occur as part of a pathway. So for example, if you had in your basic chemistry class, you had a reaction like A is broken down into B and C. Back in chemistry, you called this a reactant, called these products. Here in biochemistry, we're going to use similar terminology, but we're going to change this to substrate because we're now dealing with enzymatic reactions. So substrate is the starting compound. All reactions in biochemistry are powered or catalyzed by an enzyme. And then what's produced at the end of a biosynthesis pathway are products. But again, I will tell you that whereas you may have learned that this is true, a very simple chemical reaction can occur in chemistry, in biology, this is never an isolated event. So, for example, in this case, perhaps B and C are then further used or modified into different kinds of reactions. So, for example, maybe C is converted to D, which is then altered or phosphorylated to E. B is converted or is altered somehow to F, which is altered somehow to G. Right, so there is never an isolated event. And in fact, what I just drew for you is an example of a divergent, branched, multi-enzyme metabolic pathway where some starting compound is broken apart into two products that are then further modified to form some type of product at the end, right? Alternately, a different type of branched pathway are convergent pathways where you have some type of modification, here, another linear pathway here producing another modified product that then come together to form a complex molecule here. And these would be examples of biosynthesis where you're bringing two things together. This might be an example of catabolic reactions where you're breaking apart a molecule and releasing energy here. We'll also see some examples of linear pathways. This is an example of this one is glycolysis. There are many examples of linear pathways, but in this case, there are modifications that occur each step along the way. So this is some kind of modified product of A. So like a phosphorylation is a really common example of modifications where a phosphate group, a phosphorus with a bunch of oxygens on it, is a phosphate group. And this can be added or removed from different chemicals here. So this would be kind of an example of how A might be modified in this reaction, right? Or carboxylation. So there are different types of chemical groups that can be added or removed from molecules in each of these steps in these linear pathways. But when you see linear, you just think modification. So something's happening to this, like a chemical group is being added or removed from these different compounds here to generate this linear modification. Versus divergent, you have an actual bond being broken here. When you see two arrows coming off, that means these are both products of this reaction. Or alternately, here we have a bond being formed between C and Z to make a new product that is a combination of both molecule C and molecule Z, right? So these are different types of multi-enzyme systems where there are modifications that are occurring, bonds being formed and bonds being broken. Here in the center, a cyclic reaction. Krebs cycle is the best and the only example that I know of very well that is a cyclical reaction where some input, right, some compound comes in the Krebs cycle. This is acetyl coenzyme A, or an acetyl group comes in, is added to a chemical, and this reaction here produces a product, W, that is then modified. It's modified again. Perhaps there is an actual bond being broken here. We have a release of a product. And then another chemical reaction here converts Z back to V again. And as long as there's continuous input of U, then you have this enzymatic reaction that can continue. And so this is a cyclical reaction. And the reason we call this multi-enzyme system is because every chemical reaction is catalyzed by an enzyme. So there's an enzyme here, an enzyme here, a different enzyme here, a different enzyme here, right? So each step along the way is catalyzed by a different and unique enzyme all the way through. Let's look at some of the components of metabolism, right? So we'll start by looking at enzymes, then we'll talk about energy, looking at those chemical energy sources that we talked about in chapter 6, understand the purpose and the concept behind electron carriers, and then just mention these precursor metabolites again before we get into pathways. All right. So for enzymes, the enzymes are the same thing as catalysts. So they catalyze, or they help reduce the rate. They help lower the rate. Oh my goodness. Catalysts increase the rate of a chemical reaction by reducing the energy of activation. And so what is this energy of activation? Well, let me just kind of draw you a really simple picture of what this concept is, right? So let's say that there's someone sitting at the top of a hill, or there's a big boulder sitting at the top of a hill, and there's you as a person, and you want to There you go. You want to get this boulder down the hill. Well, you know that as long as you get it moving, then you really don't need to do anything else. It will roll all by itself. And during that process, will actually generate a lot of energy, right? All that heat that's made by the friction between the rock and the ground, etc. But the question is, how do you get it to roll? How do you get it to move? Well, even in something that will generate a ton of energy as it's moving, there is a requirement of an input of energy, right? So what needs to go into this is what we call activation. Energy. Activation energy, or the energy of activation. So to activate this reaction, there is a requirement of a little bit of input of energy. Now you could try to push the boulder. Depending upon where it sits, where it's balanced, it might move, but it would require a huge investment of energy. So to help make this reaction go faster, right, it's not going to speed this part up, but to just get this reaction moving and to help make this whole process go faster, I could provide you with a lever, right? And this lever would be akin to an enzyme. Or a catalyst. Okay, so when provided with this lever, you simply wedge it and it moves that boulder much more quickly, or at least it gets it to move much more quickly, which makes the reaction happen faster. So here is our picture of our hill, right? So let's say this is our boulder. This is that initial amount of energy it takes to get that boulder moving. And then once the boulder is moving, there is a significant release of energy as it rolls down that hill. Or let's say this is a starting compound like glucose, for example, and it can, as they as the bonds are being broken in this in the multi-step reaction called glycolysis, where glucose is broken down into more simplified products like pyruvate, the amount of energy in the product, like pyruvic acid, which is a 3-carbon compound. Sorry, that should say U. This is an example of this kind of reaction here, where this is glucose. Glucose, for example, and this is 3-carbon pyruvate. We know that there is energy released in this reaction. This is a 6-carbon compound, so a 3-carbon compound, and there are bonds that are being broken here. So the relative energy of this reactant is much higher than the relative energy of this 3-carbon product. But anyway, so let's say that we are here at the top. We know that in order for this reaction to proceed, there is an input of energy that is required. Now, without an enzyme, that input of energy is potentially too high for this reaction to occur naturally in a biological system without a huge input of heat or something else to get this thing moving. So with the addition of an enzyme, right, enzymes will then reduce that energy of activation and therefore speed up this rate of this reaction. So it'll allow these systems to actually move forward. So enzymes reduce the energy of activation, right? That's what we are saying. Increases the rate by reducing energy of activation. So we'll say by lowering this is slow. activation, activate energy. Holy shnikes. All right. Enzymes are therefore essential to cell growth because without them, many, even reactions that seem like they would normally proceed, many of these exergonic reactions that release energy, even those reactions require energy of activation. So without catalysts or without enzymes, these reactions might not proceed in a biological system, or if they do, they would proceed at a rate that's much slower than what is required for cell growth. This is a super simple slide. It's just a conceptual understanding of why enzymes are needed, right? As mentioned, that energy of activation is sometimes insurmountable. Without a catalyst, the cell would need to either increase the amount of heat to increase the molecular velocity so that these chemical reactions would actually occur, which is a very harmful process. Most cells do not do that except for those extremophiles. Alternately, potentially increase concentration of reactants to increase the rate of collision. This is usually an impractical system. Sometimes the cell does this as well, but to solve all of these problems of that activation energy, alternately a catalyst is added. So an enzyme will help speed up the rates of these reactions by reducing that energy of activation. This is a picture of some of the concepts behind enzyme structure. Now I'll tell you, I won't test you on these apo versus holo enzymes, but in case you come across these terms, this is what it means. The apo enzyme is essentially the protein core. Sometimes you can say this is the protein core with a cofactor on it, but usually what we were talking about is the protein core. So that means that you were just looking at those amino acids, potentially those internal bonds that are formed here with disulfide or phosphate groups, etc., or hydrogen bonds. But it's just the protein core that's unloaded. When you add these metallic cofactors like iron or magnesium, etc., then when these metallic cofactors are bound, or the coenzymes are bound to that protein core, then we can call that the holo enzyme. That means that it's loaded with its cofactor. Now, also, sometimes you'll hear the term holo enzyme referred to an enzyme that's bound to its substrate. That's the actual chemical that it is going to help modify in the chemical reaction. So holo kind of means fully loaded, essentially. Apo is just protein core or unloaded enzyme. So note that there are various ways that this enzyme core or protein core can be decorated, and these decorations have a significant function. In fact, most of the time, it's the cofactor that actually has the functional attributes of the enzyme. So these cofactors can either be in the form of a coenzyme, which is usually a vitamin, or a metallic cofactor, which is a metal, some type of a metal ion. Like, for example, I just mentioned a couple, like iron or copper or manganese or any of these types of metals. And these things are really important in any kind of like ionic interaction or redox reaction, something like that. Versus these coenzymes can also be important in redox reactions, but also in any kind of chemical modification that's done to a substrate. So, and this picture is just to show you that it's not just one or the other. There are some enzymes that can have both coenzymes and metallic cofactors to them as well. Just to understand, and in class I'll give you a little a workout for this one to give you an understanding of how these things work, but just as a reminder, and this is from chapter 1 or basic biology or even basic chemistry, proteins are created, enzymes essentially are created from the same process. Enzymes and proteins are the same thing. So there are 4 levels of the folding structure of a protein. Primary is the arrangement of the amino acids. So when we're discussing primary arrangement, these are individual amino acids. And this arrangement is determined by DNA. So this we'll talk about in the next chapter on genetics. This is produced during the process of translation, where DNA is first transcribed into messenger RNA, and that messenger RNA is read by translational machinery, specifically the ribosome, to generate this core primary sequence of amino acids. Now, these amino acids have their secondary formations. These are alpha helices, and beta pleated pole. Sheets. These are ways these amino acids, based on their configuration, so each amino acid has a very specific R structure. That's the part of the amino acid that comes off of its basic core. And when they bind together, they form these natural alpha helices or beta pleated sheets based on their core structures. So the secondary arrangement is formed kind of based on the primary structure there. Now, once a protein has undergone these primary and secondary formations, then they are folded through post-translational modification into their tertiary structure. Now note that the primary arrangement and the secondary structures are maintained here. Here are the, for example, the beta pleated sheets and the alpha helices that are still maintained during the forming of this tertiary structure. And this tertiary structure is formed through internal bonds. I'm sorry, guys. Internal bonds. The most common types of bonds are called disulfide bonds, where two sulfur groups get together and bond, and they bond between amino acids that have these sulfur groups on them, like cysteines, or hydrogen bonds. My computer just can't quite handle these videos, man. All right, so this forms this internal bonding of this tertiary structure, but wait, that's not all, folks. These tertiary structures then come together with other tertiary structures to form the quaternary level. So this is subunits joining together. For real, come on. U, N, I, T, S, S, U. Subunits coming together to form the final protein structure altogether. So tertiary and quaternary formations, this is called post-translational modification. So after that initial transcription translation, and these things form their secondary arrangements, then enzymes come in and help catalyze the folding of this protein. So why are we spending so much time on this? Because it's important to note that enzyme structure, this really complex folding of these proteins, create these binding sites, these pockets for the substrates. And the substrate binding is essentially what the whole purpose of the enzyme is. So its modification of the substrate requires a very, very specific docking site on this enzyme so that the substrate can come and dock and interact with the vitamin coenzymes or the metallic cofactors and have their modification. So it's very, very important to note. Let's see if I can write this here. For enzymes, sorry people. For enzymes, structure is equal to function. Exclamation point, exclamation point, exclamation point. So structure is vital to the function of the enzyme because of those really specific docking stations that are formed through that folding of the protein. And so you can see how this whole thing works. These are the interactions between enzymes and substrates. So here is a substrate, right? Something that starts a chemical reaction, for example. The substrate has a very, very specific binding site on the enzyme. And when that substrate binds or docks on the enzyme, there's this perfect interaction that allows this chemical process to occur. So we call it a lock and key or a hand and glove specificity, right? With the substrate binding to a really, really specified site on the enzyme. Here's another picture of this where we have some substrate that has a very specific chemical composition and its own particular folding pattern, binds and docks at a very specific site on the enzyme called the active site. And if this is a perfect match, then we have an enzyme substrate complex wherein the substrate can now be modified through a cofactor and then a product is released. These cofactors, as mentioned, can be elements, so metallic elements like iron or copper, etc. And these can do all kinds of different things. So they can bring together the active site and the substrate. They can also do modifications of this thing like breaking bonds or helping alter electron transfers, or coenzymes, which are usually vitamins. So coenzymes are really important in modifying the substrate through the removal or addition of a chemical group. Now, what are these chemical groups we're talking about? Well, there's all kinds of them, but I'll write down a couple of examples of these chemical groups that we could be discussing here. I wonder if there's another way that I could write this. No. Okay. So examples of these chemical groups. Example. Commonly, we'll be talking about moving electrons and protons in the form of a hydrogen molecule. So two hydrogens can be transferred back and forth, and that will transfer electrons and protons because every hydrogen ion has one proton and one electron. So when you take or add a hydrogen molecule, then you are adding or removing 2 electrons and 2 protons. Phosphate groups. As mentioned, this is a phosphorus molecule with 3 oxygens on it, so it's a high energy rich compound. It's involved in so many regulatory and signal transduction pathways, so ways that signals are transmitted from one cell to another, for example, are through phosphorylation or dephosphorylation reactions. We are going to write it like this. So whenever I write a P with a circle around it, this stands for that high energy rich compound, a phosphate group with a phosphorus molecule and 3 oxygens on it. Other types of enzyme reactions might be something like a carboxyl group. Whoops, that's spelled wrong. Carboxyl. And that would be a CO2 carboxyl group. Is that right? Carboxyl. Or hydroxyl. Where's that CO? Carboxyl is CO. Sorry, guys. This is painful, isn't it? Hydroxyl group. OH. And we'll stop there, but there are lots more on this list of chemical groups that can be added to or removed from a substrate when it docks at this enzyme, right? So these coenzymes will come and dock at that same station as the substrate binding site and physically take or add, so remove or add, some type of chemical group to modify that substrate. I'd like to switch over and draw out a couple of these things, but I just don't know how quickly my computer is going to be working. So bear with me. Okay. So when we're talking about enzyme substrate complexes, there's a few things I'd like to write out here. I always draw my enzymes exactly the same way to show you a couple of key parts to an enzyme substrate complex. So this is my enzyme. Oh my gosh. This is my enzyme. So right here at this initial part here, where I have a substrate that has a really specific form here. This is my substrate. Remember, substrate is the same thing as a reactant. It's the starting compound, but we're calling it a substrate because it docks at a very particular site on the enzyme. And this docking station is called the, let's change the color, is called the active site. Docking station or docking site for the substrate. And this is, again, that lock and key interaction where there has to be a perfect match for that substrate and enzyme for that substrate to dock here at the active site. Now, next to this docking station, I've written here, and this is just in our example, I'll keep it just like this. This is a docking station for a cofactor. Docking. Site. So cofactor, like let's just say we have a cofactor that's a coenzyme. So in our example, a cofactor might be a coenzyme. And we'll see a whole bunch of examples of this as we talk through metabolic pathways, that some of these really common coenzymes are electron shuttles. SU shuttles. So they transfer electrons in the form of a hydrogen ion. So let's say this coenzyme comes and docks right here next to this substrate. Now we have this really perfect match between that cofactor that is the actual workings of the enzyme and the substrate that is ready to be modified. Now note there's one more station down here that I'll show you as we go through here. It's really important, and this is how an enzyme is regulated. This is called the allosteric site. Allo is like but other. That's my own definition. Allosteric, and steric has to do with the structure or orientation of something. So this is a site other than the active site that's on the enzyme that has to do with the structure or conformation of the enzyme. This is the allosteric site. And this is really important for binding a regulator. I don't know why these are binds an allosteric. Regulator. We'll get back to this in just a little bit. Okay. So in this reaction, in this reaction between an enzyme and a substrate, we have these things all coming together to form an enzyme substrate complex. We'll leave this allosteric site undocked at this point. Now note that during an enzyme reaction, the enzyme never changes, even when we have the binding of our substrate here. So now we have the docking of our substrate right here at this active site. And remember that we also have this cofactor that is now docked right here at the cofactor docking site. And so we'll call this whole thing the enzyme substrate complex. Enzyme substrate complex. This is where magic happens. So whatever the purpose of this enzyme is, that's what's going to be happening. In our example, remember this is only an example. In our example, we're saying that this cofactor is a coenzyme that shuttles electrons back and forth. So let's say that this guy is going to be a catalyst that helps remove electrons and protons from the substrate. So let's give a picture of the removal of these things. In what color do you guys want to do? Let's do pink, huh? So let's say hydrogen has just been removed and taken over there. And so this is an example of an oxidation reduction reaction where this guy has just lost electrons, so it's been oxidized. The cofactor has just gained electrons, so it's been reduced. And at the end of this reaction here, again, our enzyme is not changed. It looks exactly the same. But now we have a release of that product. We have a release of the substrate that has now been modified. And now it's a product. And it is missing product. It is missing protons or electrons. We also have that electron carrier. Whoops. We have the release of this cofactor, which has happened. I lost all my colors here. Where'd my color editor go? There we go. We lost our cofactor. Our coenzyme now is fully loaded with that hydrogen molecule, and this is ready to go be a part of another chemical reaction somewhere else and then come back again and get back into the game. So these coenzymes, they essentially have one function. They add or take away, add or remove some type of chemical group, and they just kind of go back and forth through the cell doing the same thing over and over and over again. These substrates come in one time, and then once they come in, they're modified and they're released as a product. And this product usually has some other function in the cell downstream. Maybe this is glycolysis, and this product is going to continuously be modified to generate more of these electron carriers and generate metabolic intermediates and produce energy in this process. Okay. And just another note here on enzymes. These are some terms for you. So we can talk about exo versus endo enzymes. This is just their location in the cell. So if an enzyme is released from the cell extracellularly, usually for hydrolysis of larger nutrients or to neutralize toxic products in the environment, these are called exoenzymes. We've done examples of this one in the laboratory already with amylase and gelatinase and urease, amylase and gelatin. And alternately, if the enzymes are produced and stay inside the cell, these are endoenzymes. These are usually involved in catabolic and biosynthesis reactions here. These questions here should be answerable to you. Why is structure important for an enzyme to function? Well, we did talk about that quite extensively, so go back over and see if you can figure that one out. What would happen if one amino acid were changed that was located in the active site on the enzyme? So this one you should, again, be able to answer. And we're kind of going back here and saying, If there was one amino acid, remember this is all just amino acids in here, one amino acid that was changed right here in this active site, what would be the consequence to this whole chemical reaction? Again, you should be thinking that structure is equal to function. So if there's a modification right here at the active site, it potentially has the ability to reduce enzyme function altogether. Because if that substrate can no longer dock here, or if it's an imperfect fit, then its interaction here with that cofactor may not occur, and therefore you may actually just not have an enzymatic reaction whatsoever, or can be reduced in its efficacy. So it's important to understand again, or to remind yourselves that everything in a biological system inside the cell occurs as a metabolic pathway, right? So for metabolic pathway, these are multi-series steps or multi-series pathways, and each of those steps is catalyzed by a unique enzyme. And because of this, every pathway has what we call a rate-limiting step, or at least one or more enzymes that can be involved in regulating this pathway and really set the rate of that pathway's progression. This video is from your textbook. I have no idea if it's going to play on here. No. So you can go back into your textbook and watch this series of biochemical pathways, or actually go into our Canvas page and you can look up this video here for biochemical pathways. But essentially it's just going to show you how one substrate comes in to this series of enzymes here. A single substrate enters into this reaction, and after interaction with enzyme 1, remember that enzyme substrate complex, it is modified and is converted into a product. And that product then becomes the reactant for enzyme 2, and once it's modified here, becomes a product, and this product then becomes the substrate for enzyme 3, binds here and is modified to form product here, and this product becomes that final substrate for this last enzymatic reaction, and there's a modification here, right? So one substrate comes in and can essentially migrate through all of these enzymes because of the modifications that are occurring as we go along. So for a biological, or for bacteria, and actually for all cells really, it is important to be able to regulate enzyme reactions. That means turn them on when they're needed, but then shut them off when they're not needed. So regulation is a really, really vital function of optimizing energy efficiency in the cell and making sure the cell is not essentially doing things that are wasteful for it. So there are a few different concepts here behind enzyme regulation. Now, some very specialized enzymes are always present. So that means the cell is always making them and always keeping them in the on position. They're always functioning. We call these types of enzymes constitutive enzymes. They're ever present, not just being made, but also being functional enzymes. A prime example of this are those that are involved in metabolizing glucose, because glucose is the starting compound for the central metabolic pathways in pretty much every cell that's out there. Whether or not glucose comes into the cell doesn't matter. These glucose metabolizing enzymes are always present. That means they're always being made, and they're always in the on position, right? So they're always being expressed, and they're always functioning. This is as opposed to enzymes that are regulated. Now, regulated enzymes can either be induced or repressed, and there are a couple of different ways that a cell has to do both of these things. Induction means that they are in the off position, right? That means usually that they are not being synthesized by the cell. And let me just tell you what this term synthesis means, because I know we haven't talked about this yet. For all things in discussing enzymes or proteins, right, it always starts with DNA. And this stuff we'll talk about a lot more in the chapter on genetics, but DNA must be transcribed I think that's an SC, scribed, or transcription, T, R, to messenger RNA, which is a transcript. And this transcript is then translated through the process of translation. Or it is translated to a protein. Oh my gosh. There we go, a protein. This process we are going to be calling gene expression. So in the DNA, it is an actual gene, an encoding region of the DNA that is turned into a protein. So when this process occurs, that should say gene. When this process occurs, all the way down, we are going to call this expression. Oh my goodness, I'm sorry. E X P This is why these videos take so long. I-O-N. Okay. So as I'm talking through this and I say enzymes can be induced, that means they can be expressed. Expression means they can go from being just a gene to being turned into a functional physical protein, right? So induction means that maybe the cell has just put a barrier on this whole process here, so there is really no physical protein present, right? And then that barrier can be removed so that the protein becomes expressed. Repression can be the opposite of that. So repression can also be at the DNA level, where the protein or the enzyme is being expressed, is being made by the cell, and the cell doesn't want that protein to be made anymore, so it turns off the synthesis of the protein way down here at the DNA level, right? That can be enzyme repression. Alternately, the protein itself, the enzyme, can just be bound to a regulatory molecule and turn off the workings of that enzyme altogether. So there are a few different ways that you can turn off the function of the enzyme or turn off the expression of the enzyme altogether. You can repress the enzyme. And for induction, it's really just turning on the synthesis or the expression of that enzyme at the DNA level. So this is, again, kind of summarizing all of these things. So let's talk through the direct regulation or control. So here's our enzyme, right? This is sort of just a mirror image of what I drew. Our core enzyme with that active site that could potentially bind to a cofactor. Here's our allosteric site down here that I mentioned is important in allosteric regulation, binding a regulatory molecule. So here's our normal interaction where we have a substrate that binds and docks with a perfect fit at the enzyme's active site. Now in direct regulation, where we're interacting with the enzyme directly, one way of controlling enzyme activity, there is either competitive or non-competitive inhibition. So competitive inhibition means you have a regulatory molecule that competes for the active site. We call these mimic molecules, where they look a whole lot like the substrate, and they don't have the same perfect fit, but they do bind to the same active site, and they compete for binding, which inhibits enzyme activity. Alternately, we can be working with a allosteric regulatory molecule through non-competitive inhibition, right? This is when an allosteric regulator comes and binds at that allosteric site on the enzyme. And as mentioned, allosteric means a site other than the active site that has something to do with steric or conformation. So when you have binding here at the allosteric site, binding induces a physical change to the enzyme structure. And again, what do we know about enzyme structure? Structure equals function. So when you're changing the structure of the enzyme, you can lose the function of the enzyme because now the active site physically can no longer bind to that substrate. So there's a few different terms we put here to non-competitive inhibition. This is also negative feedback regulation, where the product binds to a regulatory site. And I think I have a picture of that one on this next slide. So let's look at what this concept of negative feedback inhibition is, and then we'll draw out some of the stuff too. Right, so this is feedback regulation, this concept here of, and we're looking at negative feedback inhibition. So let's say, for example, I think the best example of this is when, let's say that this is a biosynthesis pathway and the product is no longer needed because it becomes available in the nutrients. So let's say that you're growing this organism in a broth culture, and then you go and add more nutrients to the broth culture. So it originally was making one particular amino acid, and then suddenly that amino acid is no longer needed because you've just supplied it in the media. Okay, so let's see what happens here. So normally, let's say we have a substrate that then binds to the active site on enzyme 1, and there's some intermediate product that's made. This becomes the substrate for enzyme 2. There's the product, becomes substrate for enzyme 3, and here's the end product. And again, let's just say this is some particular amino acid. So the cell will just continue to work through this pathway, generating those products as long as they are needed for biosynthesis or whatever that next step is. But let's say that product becomes excessive, so you've just supplied it in the media and the cell no longer needs to make it. So it builds up because it's not being used. Well, when you have buildup of a product, that means that you are inefficiently using this metabolic pathway, and you can shut down the pathway because you don't need that product. And so the way this happens is that end product comes back and binds to a regulatory site on enzyme 1. Right? It binds to an allosteric regulatory site on enzyme 1, changes the conformation or the physical structure of this enzyme so that the active site can no longer bind to that substrate. And when you inhibit this enzyme number 1, you're inhibiting everything downstream, right? So it regulates both the enzyme and the entire metabolic pathway by blocking just enzyme 1 in this reaction. This is a picture from your textbook showing you these two things. Again, competitive inhibition is easy to understand. Here's your physical enzyme, and you have a mimic molecule that has a very similar shape to the normal substrate. They both compete for binding, and when that mimic molecule or competitive inhibitor binds, the reaction is blocked simply because that substrate cannot bind. For non-competitive inhibition, here it's a little bit more complex, whereas normally you have the binding of the substrate here to the active site, but perhaps if that product gets built up in the cell, the product of that metabolic pathway, the product can come back and bind the allosteric regulatory site, changing the conformation of the active site so it no longer binds to its substrate. And while that regulatory molecule is bound, you are shutting off that enzyme pathway by turning off the enzyme's function altogether. Stop here. So this is to continue the discussion of enzyme regulation. Let's write that down. Enzyme regulation. So there are two primary means a cell has to regulate enzymes. This is either direct regulation by interacting with the enzyme itself. Right, this is my picture of the enzyme with that active site and that allosteric site. So for direct enzyme regulation that we just mentioned, if there is a competitor molecule that binds to the active site, this is called competitive inhibition or regulation. Right? And again, this is with a mimic molecule. that competes for the active site. So here we have our enzyme, and normally we have our substrate binding. Sorry, I should have drawn that a little bit lower. Let's see if I can. Yeah. So here we have our, I think that's still on the screen, enzyme. Whereas normally we have our substrate. That should say substrate. Able to bind. Here we have a competitor that binds. Right? This is the regulatory molecule, inhibitor. That binds at the active site to prevent the substrate from binding. Now, alternately, there can be indirect inhibition or non-competitive. Inhibition. This is where a allosteric regulator binds to the allosteric site and changes the conformation, which is the shape or the structure of enzyme, so that the active site does not bind. Right, and for non-competitive inhibition, we can draw our enzyme here that normally binds. I think I just lost my red pen. Excuse me. Normally binds its substrate, but when an allosteric regulator allosteric regulator comes and binds this allosteric site, it changes the conformation of the enzyme so that it no longer binds the substrate. Now, I've mentioned already, this is a form of, usually a form of feedback inhibition, negative feedback. Where the product of the pathway is the regulator. Right, so we've talked about this already, that in this metabolic pathway, at some point downstream, this product is generated, and when that product is no longer needed by the cell, it can come back to the first enzyme or one of the rate-limiting enzymes in this enzymatic pathway, bind allosterically, change the conformation so that this enzymatic reaction cannot proceed, and thereby it shuts off its own production. Now, it's important to note this is a reversible, actually both of these are reversible reactions, because at some point perhaps that cell might need the product of that enzymatic reaction again, and so it wants to be able to turn on and off this pathway at any given time. Now I'll finish this up by looking at the other two types of enzyme regulation. That would be looking at synthesis level control, which I'll go over the slides in just a minute. So direct means interacting directly with the enzyme, or indirect, which is synthesis. This means gene expression, right? Gene expression. So this is regulating the expression of the enzyme at the DNA level. Now from here, there are two ways that you can consider this. That would be either turning on or turning off enzyme expression, right? And remember, expression means from DNA to mRNA to protein, so transcription and translation. So you can have an enzyme that's always turned on, and then these guys can be turned off, right? To turn an enzyme off, that means you are repressing them. So these are usually expressed, usually on, and you can turn them off or you can repress them. These are enzymes that are usually involved in biosynthesis. And because of this, usually repression is also a form of negative feedback inhibition. Where the product of the pathway is the repressor. Now remember, this is at the DNA level, and so when we get into our next chapter, we'll see exactly how this process occurs. But the product essentially comes back all the way around to the DNA at the gene level and turns off the ability for the enzymes involved in this pathway to be synthesized at all, and therefore the whole pathway gets shut down altogether. This is as opposed to enzyme induction, repression and induction. For induction, to induce means to turn on, right? So you're turning them on, which means that these guys are usually not expressed. Usually in the off position, or they're not expressed. You can turn them on. Sorry, you're seeing my living room in the back. These are processes that are involved in catabolic reactions. Catabolism-based enzymes, or breaking things down. A really classic example of this, which we'll look at in detail in the next chapter, is the lac operon, which gives the ability for the cell to use lactose. And these reactions are shut off unless the cell actually needs to use that particular substrate. So usually for catabolism, as mentioned, glucose metabolizing enzymes are always turned on, so those are constitutively expressed, and only when glucose is absent and the other substrate is present will the cell turn on these inducible operons, these catabolic operons. So in this case, the substrate for that metabolic pathway is the inducer. And I'll say usually glucose must be absent. For these inducible operons to be turned on. Oh shoot. Still recording? I think it is. Sorry. Usually glucose must be absent for these inducible operons to be turned on. Again, to enhance your understanding of synthesis level regulation, we'll get into this in a lot more detail in chapter 8, but just a basic concept here. Here's our DNA, here's our genes that are encoded on our DNA. And let's just talk about prokaryotic cells here, because for eukaryotic cells, it's a little bit more complex. Here is the regulatory locus, so at the beginning of every gene segment. And let's just do this example that we'll go over again in our next chapter. Prokaryotic cells like to stick similar genes together that function in the same metabolic pathway. So for example, in this case, this is an example of the lac operon. I won't go into all the detail here, but when the cell needs it, when the cell has lactose present, these genes will be expressed, and that means transcribed and translated into different enzymes involved. Enzyme 1, enzyme 2, enzyme 3, right, that are all involved in breaking down lactose, so that when lactose is present, lactose becomes the substrate for enzyme 1. It's not exactly true, but we'll just kind of go with it here. Actually, enzyme 1 brings lactose into the cell. Lactose becomes the substrate for this enzyme here, and a product is produced that then becomes the substrate for enzyme 3. Whoops, that one doesn't work. And there's a product that's produced here that includes glucose that can then go work in the cell. So this is the catabolic reaction where lactose is brought into the cell and then is broken down into a usable form. So when glucose is present, glucose is preferentially used in the cell. So the cell doesn't want to turn on this machinery that requires the input of energy in order to move forward with this reaction. So when glucose is present, and especially when lactose is absent, then this whole region of the DNA is essentially shut down, right? So under normal circumstances, when glucose is present and lactose is absent, then this whole thing is shut down. It is repressed. Repression is where there is a blockade to this entire thing. So there is no synthesis of the genes on this DNA. That's repression. So none of this stuff happens. Therefore, there is no reaction here because there are no enzymes for this reaction to occur. Now, when lactose is present, lactose is the substrate, right? For both enzyme 1 and enzyme 2. So when this lactose is present and when there's no glucose in the cell, lactose will actually remove this repression, and it will induce this whole synthesis of these enzymes in this reaction, right? So this is induction, where the substrate comes and turns on the expression of the enzymes here by taking away anything that's repressing this set of DNA. The opposite would be true when we mentioned enzyme repression. Under repression circumstances, everything is being produced and then it can be shut down with the presence or with the negative feedback inhibition. Again, in continuation of enzyme regulation. So as we have already mentioned, enzymes can be regulated directly by binding either competitively at the active site by a mimic molecule, or binding allosterically, usually with the product of that enzymatic pathway coming back to shut off that pathway. Now, in addition, enzymes can be regulated at the synthesis level. This is the process from DNA to RNA. This is transcription, and then to protein, which is translation. Remember those post-translational modifications I mentioned, so this thing needs to be folded up. And this is the enzyme. The enzyme reacts with a substrate to produce some kind of a product. This can either be where that product becomes just as we mentioned before, so perhaps this is an amino acid synthesis metabolic pathway where that amino acid is no longer needed because it becomes available in the environment, at which case that product can then go back all the way to the DNA level and essentially turn off the expression of the genes in that area of DNA to eliminate the enzyme itself, right? So that's turning off enzyme synthesis, which is enzyme repression. The products of that enzymatic pathway or enzymatic reaction will go off and turn off the transcription or be the repressor. This is as opposed to enzyme induction, right? And again, we mentioned already that for each of these things, enzyme repression that we just looked at a second ago, enzyme repression happens where these are anabolic reactions usually. Biosynthesis. Where the product becomes available, or maybe that cell is becoming in that starvation mode and is shutting off biosynthesis pathways. So in either case, you'll have a buildup of the product, and the product can come back and repress the synthesis of the enzyme. Because this is another example where products come back, we know also this is negative feedback. Negative feedback regulation. Versus enzyme induction, the synthesis of the enzymes is induced by the substrate of the enzymes, right? Synthesis of the enzyme is induced by the substrate of the enzyme. In other words, when the enzyme is needed because the substrate is present, then the enzyme will be made, right? When the enzyme is needed because the substrate is present, then the enzyme will be made. These are almost always catabolic. Catabolic pathways or enzymes, right? This is the example that I gave you, the lac operon. There are many other examples too, but this is a really classic example. Under normal circumstances, these guys are turned off, right? These guys are normally off, and they can be turned on. Up here for enzyme repression, these are normally in the on position, and they can be turned off. So here for enzyme induction, these are normally in the off position because the genes are repressed from being expressed. So there's a whole set of regulations that are occurring. There's a repressor that just sits upon the DNA and prevents the synthesis of the proteins that are encoded by these genes here. Now, when, as mentioned, glucose is absent and that substrate is present, in this case it's lactose because we're looking at the lac operon, lactose becomes the inducer of the enzymes involved in lactose catabolism. Remember that when the substrate of the enzymes is present, in this case lactose is the substrate of the enzymes involved in lactose catabolism. And so these enzymes right here, these enzymes break down lactose to produce glucose here. And so these enzymes are only turned on or made when they are needed, when lactose is present. Right? We'll look into the details of exactly how this whole process is working in Chapter 8, but for now just understand that lactose, which is the substrate for the lactose catabolizing enzymes, lactose, the substrate, will turn on the synthesis of the lactose catabolizing enzymes, right? So lactose is the substrate, and it induces the synthesis of the lactose metabolizing enzymes. So this is going back to a concept we talked about before, right? From Chapter 6, we talked about how The sensitivity to environmental changes, including pH, temperature, different types of chemicals, saline, etc. All of those environmental sensitivities are based on enzyme activity. So enzymes will operate under the organism's habitat conditions. There's a very narrow range for each species for their enzymes optimal activity. And this is oftentimes due to denaturization that can occur at extremes or changes in these different conditions, like high temperature, changes in pH, high or low, and there are certain chemicals that can also cause this. Denaturization means breaking bonds on the inside of a molecule, and that unfolds that molecule essentially. For proteins, remember that those internal bonds are really important excuse me, in the tertiary and quaternary structure of these proteins. And so when you're breaking those internal bonds, you're unfolding the protein's structure, and therefore you're eliminating the enzyme's function. So we talked about how allosteric regulation is a reversible reaction and the importance of understanding this. Remember that allosteric regulation is negative feedback inhibition, where a product of a biosynthesis pathway is no longer needed by the cell, and that product goes and shuts down its own synthesis. So it's important that it's reversible because when the environmental conditions change again, perhaps that nutrient becomes depleted in the environment, that cell must be able to turn back on the synthesis of that nutrient so that it doesn't die. What is the function of a coenzyme? I think I'm going to let you answer that one. You can go back through and understand what those coenzymes do. Oh, and that's it for those discussion questions. Okay, let's finish these sets of slides by talking about energy and some precursor metabolites that are generated. So as an overview, the cells obviously need energy for metabolic processes, specifically for biosynthesis, building up those larger components of the cell so that the cell can then divide. We've talked about this from chapter 6. Cells can get their energy through sunlight-driven reactions, specifically photosynthesis, or through catabolic reactions of organic compounds. Let's do a quick step back from our last chapter. Remember these prefixes. When we were talking about the source of energy being light, we were talking about these guys being phototrophs, right? Versus if they undergo catabolic reactions to get their energy, these are either chemo, organotrophs. Remember that they use organic compounds for their energy source, or chemolithotrophs, where they use inorganic compounds as their energy source. So now we'll look at how these processes occur, and one of the biggest differences is in the end electron acceptor for these different guys, so we'll see how some of this stuff works. Energy is stored in the form of ATP. We call this the energy currency of the cell, and anytime energy is used, it must be replaced. So we'll see the ATP process, or the cycle. This is just a quick overview or a reminder, essentially, from your general biology class. The majority, not all, but the majority of the energy we have on our planet comes from solar energy, from energy from the sun. Comes into our planet and is converted from light energy into the form of chemical energy, glucose specifically, through the process of photosynthesis, right? So photosynthetic organisms like our producers here have seen all of our plants, but also remember we have some photosynthetic prokaryotic cells that can also photosynthesize and generate chemical energy from sunlight. Anyway, these are our producers. They produce chemical or usable chemical energy that then everyone else on the planet, chemoorganotrophs, consumes that chemical energy and breaks down the bonds here to generate ATP. And this process releases carbon dioxide and releases water in the process here. and there's a whole cycling of these different chemicals on the planet. These are different terms for you: exergonic versus endergonic. Exergonic reaction, don't get too confused with exoenzymes and endoenzymes. Those are different concepts altogether. Exergonic reactions are reactions that release energy in the process. Here is a picture of a complex molecule that when the bonds are broken and there are simpler molecules generated in this reaction, there is a release of energy. So when bonds are broken, there is a release of energy. Endergonic reactions are ones that require the input of energy or driven by the addition of energy. So here, where a bond is formed between two simpler molecules to generate a more complex molecule, this is an example of an endergonic reaction. And you should look at these and be thinking catabolic versus anabolic, which is which. Since this is forming a bond, since this is breaking a bond and releasing energy, this is a catabolic reaction or catabolism. Since this is the formation of a larger molecule from two more simple molecules and a bond is formed, this is anabolic reactions. Or biosynthesis. So ATP is, again, the energy currency of the cell. This is adenosine triphosphate. It's a type of a nucleotide. Is a ready, immediate donor of free energy because of the strain between the third and the second phosphate groups, right? These phosphate groups are, as mentioned before, high energy-rich compounds. They have a very strong negative charge to them because of all those oxygen molecules. So when you put two of them together, they really, really repel each other. Because of that, there's a huge amount of potential energy between the third and the second phosphate groups. And when that bond is broken between the second and the third, there's a huge release of energy. Here's this picture of how ATP, or what happens when a phosphate group is removed. So this is adenosine triphosphate, T is for 3. When a dephosphorylation reaction occurs or phosphate is removed, there's a huge release of energy here, and the resulting product is adenosine diphosphate. Now, under most circumstances, the cell will cycle back and forth between ADP and ATP. But under starvation conditions, there are occasions where ADP can further be converted or dephosphorylated to adenosine monophosphate with the release of a phosphate group, but there is much lower amount of energy produced here. In fact, this is so rare in the cell that when AMP is produced, it's a sign that the cell is in starvation mode, and it triggers a huge change in gene expression so that they can start producing energy from different compounds. This is a picture of what this looks like. Here's the adenosine component, where you have an adenine nitrogenous base bound to a ribose sugar, very similar to what you would see with a nucleotide in DNA. Here is the phosphate group, so a phosphorus molecule bound to 3 oxygens, another phosphorus bound to 3 oxygens, 4, and then another phosphorus here. So these phosphate groups are huge energy-rich compounds, and again, there's a lot of repellent forces here between these phosphate groups here because of those negatively charged oxygen molecules. So as already mentioned, the biggest potential energy occurs right here between the third and the second phosphate groups. So when this bond is broken, there is an immense release of energy. When this bond is broken between the second and the first, there is still a release of energy, but it is much lower potential energy as compared to the release of the energy here. As mentioned already, ATP, when it's used, it must be replaced. So there are a couple different ways that the cell has to do this, but let me just take a really brief segue here and show you what we are talking about. So I'm gonna start a new little image here. Sorry, let me save this for you. Okay. So we are talking about ATP. So when ATP, adenosine triphosphate, and actually let's make this really bright red, right? This is a huge energy-rich compound because of those 3 phosphates. 1, 2, 3. And the biggest potential energy is right here between the 2nd and the 3rd phosphates. ATP can be dephosphorylated, right? So a phosphate can be released in this reaction. This also induces a huge release of energy to form this intermediate that's adenosine diphosphate, ADP, which still has potential energy but a lot lower than ATP. So this now has 2 phosphates on it. So in the cell, usually the cell cycles between ATP, the release of energy through the loss of a phosphate group, and ADP, and this is then, there's a reaction called a phosphorylation reaction to add a phosphate group back on. So I'm going to write phosphorylation here. Phosphorylation is the process where a phosphate group is added to a compound, right? So ADP is phosphorylated to make ATP, and the opposite is true here. This is called dephosphorylation. Now we'll just note again, this is the ATP cycle in the cell. ATP is dephosphorylated to make ADP. This also releases a huge amount of energy, and I'm just going to demonstrate to you that oftentimes this reaction, ADP to ATP to ADP, helps move forward some type of biosynthesis reaction. So let's just say like X plus Y makes Z, but it is powered by this dephosphorylation reaction, right? This is a biosynthesis. Oh my goodness. Biosynthesis. So we know that it requires the input of energy, and the energy here, comes from the release of this bond here between those phosphates. That energy will catalyze this, will help catalyze this reaction to make these biosynthesis pathways work, right? So this is just an example of when you would see this whole process, ATP to ADP. There's a huge release of energy, and that energy is captured in these biosynthesis pathways or other processes in the cell. But the result is this lowered energy compound, ADP, and this needs to be phosphorylated to make more ATP so that these other pathways can continue. Now, as mentioned, there is another, what color am I going to move to? There is, let's go to orange here. There is another pathway that can occur, which is dephosphorylation of ADP to AMP, right? Which is a much lower release of energy. So there still is energy that's released here, but it's much lower. And this process usually triggers kind of survival mode. Where it really changes gene expression, usually shifts metabolism, because it tells the cell that it's starving, that it's about to die, because there's no more generated ATP, there's no more phosphorylation reactions to make ATP. So the cell is about to die unless it does something different. So it triggers this really big shift in the cell, This is true in our cells too. In fact, AMP, or cyclic AMP, if you may have heard of that, is a huge trigger for changes to our cell, because it signals exactly this, the depletion of energy-rich compounds, and the starvation of the cell. So AMP can be rephosphorylated too, right? So it can be phosphorylated back to ADP and then back over here to ATP. If the cell, if the nutrients change or the environment changes, then there can be essentially come back from survival mode back into active metabolism. But this is the ATP to ADP cycle, and this is really what we're focusing on. So there are different ways that the cell can undergo this process here, this phosphorylation, which is what we're talking about here, right? When ATP is used, it must be replaced. In other words, when there's a ATP dephosphorylation and release of energy, then that ADP, adenosine diphosphate, must be phosphorylated to make more ATP. In the cell, there are two primary means by which phosphorylation can occur. The first is the easiest to understand, where a substrate that has a phosphate group on it donates that phosphate group to ADP to make ATP. And this is called substrate level phosphorylation, right? So substrate has a phosphate, it donates it to ADP to make ATP and a dephosphorylated substrate. Now, just to confuse you even more, this is a chemical group, right? A phosphate group. And because we were talking about removing a chemical group from a substrate, please note that there is an enzyme, and specifically a coenzyme, that's involved in this reaction that allows the transference of this phosphate group from the substrate to an ADP molecule. But this is substrate level phosphorylation. As we go through our metabolic pathways in the next sets of slides, we'll see specifically sites at which substrate level phosphorylation occurs to generate ATP. This, however, is not a big producer of energy in the cell. In fact, in the cell, cells that conditions that require a very rapid output of energy will do this over and over and over again with the process of fermentation. Our cells do this too. For example, when you work out a lot and you deplete your glucose or your energy stores, your cells will then release glucose into, your cells will then utilize glucose and convert them rapidly into lactic acid, releasing or producing a very small amount of ATP through substrate level phosphorylation. But just to give you an example of the poor energy efficiency of this process, for one glucose molecule that comes into the cell, there are only 2 ATPs, that can be generated through just substrate level phosphorylation. A much more efficient process is something called oxidative phosphorylation, and this is through aerobic respiration or anaerobic respiration, where there is complete oxidation of that initial starting compound. Again, glucose is the most common starting compound for this process here. And again, just to give you a comparison, whereas in fermentation, glucose can produce 2 ATP through substrate level phosphorylation, oxidative phosphorylation can generate upwards of 36 to 38 molecules of ATP for every 1 glucose that starts off this reaction. Right, so a huge difference in the energy production of substrate level phosphorylation versus oxidative phosphorylation. But this process does take a little bit longer time. Okay. Oxidative phosphorylation is the process by which ADP is phosphorylated at the end of a respiration pathway using a really cool compound. This is chemiosmosis, but this is an ATP synthase. It's a super giant molecule that spans the membrane and takes a gradient of protons and allows them to facilitatively diffuse back into the cell, and essentially captures that diffusion energy to power the production of ATP. This is oxidative phosphorylation because it takes advantage, or it uses a energy gradient that's generated through reduction and oxidation reactions. That is how this proton force is generated here, this huge gradient between these protons in the periplasmic space and the inside of the cell. So this is the electron transport chain that we're talking about here at the end of either aerobic or anaerobic respiration, and it has a huge amount of energy producing capacity. There is a third process. This is photophosphorylation, where ATP is obtained through sunlight-driven reactions, but we are not going to be talking about this one at all in this semester. So if you're interested in photosynthesis and how ATP can be generated through photophosphorylation, you're welcome to look this up. Your textbook has a little bit of this stuff in it, or else you can find more information online. So those chemical energy sources that we talked about in chapter 6, right, can either be organic or inorganic compounds that can come in and be broken down. All of these things require redox reactions to break them down, or reduction oxidation reactions. So let's talk about what these reactions are. Redox is reduction and oxidation, and there's a bunch of different ways that we can describe this thing, but in this picture here, it's pretty simple. Again, the most simplistic form of this stuff is to understand the transference of electrons. So in this picture, the blue guy has an electron on it. It's transferred to the red guy. So the blue guy has been oxidized and the red guy has been reduced. And I use, everyone has their own different, what do you call this, mnemonic device, but I say Leo says Gur, and this is the loss of electrons. Is oxidation. Versus the gain of electrons is reduction. Right, so in this picture we have the blue guy has lost an electron, so it's just been oxidized. The red guy has gained an electron, so it has been reduced. You can also call this the reducing agent because it reduces this one by passing its electron over. This is the oxidizing agent, etc. So in the cell, we'll see this a whole bunch. Redox reactions are how energy is essentially generated through oxidative phosphorylation, and in the cell, as mentioned before, hydrogen, the hydrogen compound or hydrogen molecule, is used to transfer those electrons. A hydrogen atom has one proton and one electron, and a hydrogen molecule therefore has two protons and electrons. So this is how electrons are transferred in the cell. These are carried by these coenzymes that act as electron shuttles. The most common one is nicotinamide adenine dinucleotide, or NAD. You can see that this is these are the two common forms of NAD. So NAD, when it is not carrying the electrons and protons, is in this form here. So this is the oxidized form of NAD, and this is the reduced form of NAD. It's NADH plus H plus. And if you're confused on that, you can just kind of see this image here. So again, this is NAD, or NAD plus is the oxidized form, and this is how it looks. So there's two ribose sugars. There's an adenine here, a nitrogenous base, two phosphates that connect these guys together, and then this is the nicotinamide group on the top of this compound. Nicotinamide can pick up two electrons and a proton here, and then there's kind of this one proton that sort of, I don't know, hangs out. It's a really bad way to put it, but that's kind of how you can think of this thing, that proton stays associated with this compound, forming NADH plus H plus. That's why we say it like this. I'll continue to write it like this. Other people just write NADH, but it means the same thing. This is the reduced form, where two protons and two electrons are picked up from some kind of a substrate and carried, usually donated to the electron transport chain, which we'll talk about later. There are a couple of other electron carriers that are seen in the cell. We'll also see FAD, and this is the oxidized form. It can pick up two protons and two electrons and form FADH2. And then this NADP, we really only see this during biosynthesis reactions, and since we're not covering any of those in our chapter, then we'll just mention it here. But if you see that in downstream studies, then you'll understand that's also another type of electron carrier. In respiration, electrons are shuttled from these electron carriers to a terminal electron acceptor, oxygen for aerobic respiration, or an oxygen-containing compound for anaerobic respiration. And again, I think that my videos are not working here in this thing. Yep. So you'll have to go look this one up, how NAD works in the cell, and how it transfers things. Now I'll just kind of forewarn you, in this video, obviously there's a mitochondria, therefore we're talking about a eukaryotic cell, but the process still occurs in prokaryotic cells as well. So we'll go into those in a lot more detail in the next sets of slides on metabolic pathways. For now, we've just looked at the different phosphorylation reactions that can produce ATP. Hopefully you can go back and understand the significance of NAD, what that thing is, and what it does in the cell. We just looked at this, but you can review again what's the oxidized versus reduced form of NAD and FAD, and then go back and understand how enzymes are regulated. Again, the two categories of enzyme regulation. I'll give you a hint, this is direct and indirect. And then those four mechanisms, so under direct enzyme regulation, looking at both of those, competitive versus non-competitive inhibition, and then for synthesis level, looking at repression versus induction. Okay. Oh, one more thing, I see this one here, sorry. For precursor metabolites, I do want to mention this as well. I think this is probably the last one for the set of slides here. For precursor metabolites, so understand that during the process of catabolism, so we're going to get into pathways in just the next set of slides, and we'll kind of stop and highlight a few of these precursors, but these are metabolic intermediates that are generated through these metabolic pathways. And as a molecule is broken down, for example, when glucose is broken down or oxidized to generate energy, there are various steps along that pathway where these little intermediate molecules that are generated can be pulled out of that pathway and used for synthesis, so used to make macromolecules. And just concept-wise, right, any molecule that can't be synthesized from precursors must be provided as nutrients. So this is what helps establish the nutrient requirements for each individual cell. And as a reminder, we're going back to this image, right? We're going to look through these metabolic pathways where glucose is broken down to generate energy in these precursor metabolites. But understand that this is really for the purpose, for two reasons. One is to generate energy so that these biosynthesis pathways can happen. And the second is to produce these metabolic intermediates, these precursor metabolites that can be used to actually move forward with metabolism. We've seen that one already. So we're going to move into metabolic pathways next and understand, wow, I have a lot of things in here. Metabolic pathways, and understand how these catabolic processes can generate energy in the form of ATP. So we're going to pause here and pick back up again in the next set of slides with catabolism.